Do you know, in addition to normal respiration, there are certain plants which also require additional respiration? No, not all the plants, few plants. And that respiration is actually called as a photorespiration or light dependent respiration. This is Dr. Pankaj Kumar, your mentor, and today we will be talking about the photorespiration, right? So photorespiration is a light dependent respiration, okay? And three organelles are involved in the process. They are chloroplasts, peroxisomes and mitochondria, okay? And here what happens that Rubisco shows oxygenase activity. Mind it, oxygenase is not an oxidation. That is actual a different behavior of Rubisco enzyme under different conditions, okay? Under high CO2 concentration they shows carboxylase activity, and under relatively higher oxygen they shows the oxygenase activity, okay? So what happens here, the 5-carbon compound Rubisco splits into two molecules, one of 3-carbon compound, another of 2-carbon compound. The 3-carbon compound is actually 3-PGA and 2-carbon compound is a phosphoglycolate, okay? And rather phosphoglycolate gets converted into glycolate which is going to act as a substrate for the photorespiration, right? So just have a look at this entire cycle. So this is a Calvin cycle, this is a RUBP, and under high oxygenase activity it is splitting into the 2 compound, as I told you, that is the 3-carbon compound 3-PGA, 2-carbon compound phosphoglycolate, right? Phosphoglycolate is getting converted into glycolate, okay? And this glycolate is going to act as a substrate for photorespiration, and this will eventually go to peroxisomes for the next set of actions. Have a look at peroxisomes. So in case of peroxisomes, what happens? Glycolate gets converted into glyoxylate. Mind it, this is the most important event. Why? Because this is a step where oxygen is actually being consumed. So here what happens, that in the presence of enzyme glycolate oxidase, oxygen being consumed and glyoxylate is formed. And as a result, as a byproduct, hydrogen peroxide is formed, which is a very toxic. So it immediately need to be broken down. And it is actually done by the enzyme called catalase. Okay, so that's why the catalase is found in peroxisomes, and that is very, very important and the most active enzyme. Okay, so it immediately breaks the hydrogen peroxide into H2 and oxygen. Now glyoxylates actually gets converted into glycine, right? And that is going to occur in the presence of enzyme what we call glutamate glyoxylate aminotransferase. So that is a step of transaminase, okay? And now the glycine is actually transported to the mitochondria. Mind it, glyoxylate, glycolate and glycine all are the 2-carbon compounds. So they migrate to the mitochondria, and what happens, that 2 molecules of glycine together form 1 molecule of serine, right? So you can see there said this is a 3-carbon compound and this is a 2-carbon compound. So altogether what we see, that out of this 4 carbon, 3 is actually getting utilized for the formation of serine, and another 1 CO2 is actually released. Now serine again goes back to peroxisomes, where under the enzyme what we call serine glyoxylate aminotransferase, it is formed hydroxypyruvate, and hydroxypyruvate let's get it converted into 3-PGA, right? And 3-PGA finally goes back to the chloroplast, okay? So this is how the entire Calvin cycle is actually getting completed, right? So let's see the entire step once again. What happens? Glycolate is transferred to peroxisomes, right? In peroxisomes, it binds with oxygen to form glyoxylate in the presence of enzyme called glycolate oxidase. So glyoxylate is aminated to glycine, and finally glycine is transferred to mitochondria, where 2 molecules of glycine together form 1 molecule of serine, okay? And serine finally in the form of 3-PGA is transported back to the chloroplast. So this is all as far as the entire photorespiration, okay? Now one questions. Photorespiration is negligible in C4 plants. What is the reason? You see, in case of C4 plants, we have seen that carbon dioxide is taken to the Calvin cycle through the malate or in the bundle seat. So what happens, that the bundle seat do not allow any access to the Rubisco of oxygen. Oxygen is not there. So that's why it undergoes only carboxylase activity, not the oxygenase activity, because right now we have seen that dual activity of Rubisco enzyme is only due to the exposure of the different the concentration of CO2 or O2. Okay, so it is the Cranch anatomy which ensures that the photorespiration is negligible in the C4 plants. Now, what are the advantages of photorespiration for plants? Well, normally it looks that photorespiration is going to have some sort of degradation of the photosynthetic yield only, but that is not the case. You see, photorespiration is a source of important metabolites like serine, glycine, hydroxypyruvate to the plants. Okay, so through that these intermediates are formed which participate in some other biochemical activities, right? So this is all what I have to discuss. Thank you.